Okay, hello. This is a lesson on COVID-19 vaccines. I kind of want to explain how the vaccines work here so you have an idea of what you're looking at. All right. So the scientific name for the COVID-19 virus is SARS-CoV-2. All right. The term SARS stands for Severe Acute Respiratory Syndrome. All right. For those of you who don't know the word acute, that means rapid onset. Hit you fast. All right, so the CoV part stands for coronavirus, and coronavirus is a group of viruses that primarily infect the respiratory tract. In fact, one example is a common cold, and this is kind of what they look like. All right, here's the virus, and these little dots around it look like halo there, right? And maybe I think that's why they call it corona, like a crown, right? Corona means crown, and those are actually spike proteins which attach to human cells. We'll talk about that. All right, so the outline of what I want to do here is, the main goal is to teach you about these vaccines, right? But I want to give you a little background, so we'll review some of the COVID-19 basics, and then explain how viruses infect people, and then explain how your immune system fights off viruses and other pathogens, which cause you to get sick. Examine different types of vaccines and how they work. All right, that's the main goal here. All right, so COVID-19 virus basics. All right, so COVID is a virus, and viruses are one of four main categories of pathogens. The word pathogen, pathos means illness, gen means generator, so these make you sick. Pathogens make you sick, all right? All right, you have viruses, you have bacteria, fungi, mold, and you have parasites, all right? So the infection transmission of the COVID-19 virus is via aerosolized respiratory droplets formed when an infected person coughs. And when you say aerosolized, that means that these float in the air and stay in the air for a while. All right, they're tiny little droplets. All right, so when they cough, sneeze, talk, breathe, whatever. And the range is between 6 to up to 16 feet away they can travel. All right, and time airborne is up to 3 hours, depending upon whether or not you're inside and the amount of ventilation. All right, okay. So infection site is primarily the lungs via the ACE2 receptor located in the lungs. And I know this is a fancy term, and you're probably thinking, what the hell is this? But I'll explain it later. All right. All right, so symptoms. First of all, you get a symptom delay due to an incubation period that takes between 2 to 14 days for symptoms to appear. All right, and that makes it kind of tricky to determine exactly when and from whom you got this from. All right. So early symptoms include loss of taste or smell. So you can't taste your food or smell things, and that's kind of early indicator. And the follow-on symptoms are you have like cold-like symptoms, like a cough, runny nose, sore throat. You have flu-like symptoms, things like fever, chills, tired, weakness, muscle aches, headaches. Those are more like flu stuff, right? And then you can have severe symptoms like shortness of breath, difficulty breathing, chest pain. The severity of symptoms range from asymptomatic to mild symptoms to severe. All right. The severe symptoms appear to be caused by a hyperinflammatory immune response due to something called a cytokine storm. It's a big fancy term, so you have way too much inflammation. Basically, your immune system is overreacting, all right? And that's due to this cytokine storm. Now, cytokines are chemical signals, right, released by certain cells in the immune system that say, hey, you guys, get your ass in gear. Let's go fight this thing. Right? Now, if you release too much, then you have an excessive immune response, and that results in too much inflammation, which then can destroy things like alveoli, which are the tiny little air sacs, that form your lungs. I mean, you got millions of these. If you destroy these, well, you're not breathing, right? And okay, so this increases risk of hospitalization and death. So risk of factors for mortality are increased age, if you got pre-existing lung problems, pre-existing heart disease, diabetes, obesity, cancer, blood disorders, a weak immune system, chronic kidney disease, or chronic liver disease. All these can make you have, increased risk for, dying. All right, so now the second goal is to explain how viruses infect people. All right. So viruses are not considered living organisms because they cannot reproduce themselves. Right? To reproduce, viruses must invade a human cell, then hijack the cell's manufacturing system to make proteins and nucleic acids to form new viruses. So see the diagram. Here you have COVID-19 virus. Right? That's a virus. These are the spike proteins, right? And it's binding to a human cell. So the blue thing is a human cell. All right? When it binds to a human receptor, this is a receptor protein on the surface of a human cell. The function of these receptors are to pull things in that bind to it. So it's going to pull in the virus. All right? So that's how the virus gets inside the human cell. Alright, so viruses are coated with spike proteins that act like hooks that bind to human cells. Alright, the spike protein on COVID-19 binds to a surface protein on a human cell called ACE2 receptor. Alright, right there, that little green thing. Alright, now the real function of the ACE2 receptor is to bind to a chemical in your body called angiotensin tensin 2, right? And so it binds it, and then it pulls that necessary chemical inside the cell, right? So basically, the COVID viruses, mimic angiotensin 2 and fool the ACE2 receptor into pulling it inside. Alright, so once bound to the ACE2 receptor, the receptor pulls the virus inside the human cell. Once inside the human cell, the virus releases its mRNA, which is its genetic material, and it's a genetic code to make more viruses. Then the mRNA goes to a ribosome, which is the protein factory for the cell, and the ribosome then makes more COVID-19 viruses. So that's how viruses invade and hijack a human cell to reproduce and proliferate. And that can then impair bodily function, cause disease. Right? So here's the inside of a human cell. So the pink stuff is a human cell, right? And then if a virus gets inside, what it does is it's going to release its genetic code, right, to make more viruses. That's the mRNA. And that goes to a ribosome. Ribosomes are the protein-making factories of the cell, and it kind of goes through there, reads a code, and based upon the code, makes the proper proteins. Now, so what it does is it makes more viruses. All right. Now the human cell will take a fragment of the virus, which is called an antigen. So it's called an antigen because it generates an antibody response, and an attack upon it. Right. So here's the antigen. It's a fragment of this, and what the cell will do is it'll place it upon a molecule called a major histocompatibility complex type 1. MHC1, major histocompatibility complex type 1. I know it's a mouthful, right? All right. So now normally major histocompatibility complex type 1 by itself, here's by itself. This is an ID. This is like your ID badge. It's like, hey, it's me. I'm one of you. Don't kill me. Right? This is IDs as self. It's a self ID. Analogy can be like a U.S. flag in a war. You wave the U.S. flag, hey, don't shoot me. Right? I'm one of the good guys here. I'm one of us. But a major histocompatibility complex type 1 combined with an antigen marks the cell as infected and to be destroyed. Analogy is a flag upside down. right? So this is, hey, don't shoot me, I'm one of us. This is, warning, danger, kill me, right? So it's kind of like a suicide signal. It's like, I'm a goner, kill me. All right, so, okay, all right, so yeah, you're right. Yeah. I'll explain this more of that in a second. All right, so it sends a signal to other immune cells to come along and kill it. So COVID-19 virus has a spike protein that binds to the ACE2 receptor to enter human cells. The ACE2 receptors are located throughout the body, but they're especially prevalent on the lungs and small intestines, and that's why you get these respiratory problems. All right. So now this is how your immune system fights viruses. This is the third goal here. Okay. All right. So immune system, it fights pathogens. And again, pathos means illness, gen means generator. So these are foreign things that get inside you and they cause disease. Four main categories are viruses, bacteria, fungi, mold, and parasites. All right. Now, your immune system can be broadly divided into two main parts. One is innate and one is adaptive. The innate part of the immune system is always turned on. They're always on guard and they give you an immediate response, right? They're always protecting you. Now, the adaptive must be turned on, right? This is kind of like the cavalry that comes to the rescue, but it takes about 4 days to respond, right? And that's probably why colds and flus last about 5 days. Because by the fourth day, these guys kick in, and then these guys are like heavy hitters, and they'll whack that cold or flu by the fifth day it's over, right? All right. Okay, some examples of your innate immune system, which is always on, is skin. I mean, you always have your skin on you, or acid secretion, like in your stomach. It's always producing acid. And some more important cells are these natural killer cells and macrophages, and there's a whole bunch of other ones, which I'm not going to explain. I'm just going to focus on these two. So you can, if you're interested, you can look these up yourself. All right, they play a big function, but I'm going to focus on just these two. So the natural killer cells are these here, all right? And I say, well, you can think of these as being like cops, right? That go around and checking IDs on other cells, right? So the two main ideas are the major histocompatibility complex type 1. Remember I told you that's like an ID. Hey, don't kill me, and the other is called an activating ligand. So the major histocompatibility type 1 is an ID as self. Don't kill me, I'm one of us. And the activating ligand is like suicide by cop signal. All right. So here's three situations. You got a healthy cell, you got a foreign cell, and you got an infected human cell. Right? So on a healthy cell, you have an even balance of major histocompatibility complex type 1, the self-ID, along with the activating ligand. And when it's a balance, the natural killer cell, which is the cop, comes along and says, okay, you're good, all right, I'll leave you alone. Right? But in a foreign cell, it's not going to have that major histocompatibility complex type 1 at all, right? So it says, okay, you're a foreign, I'm gonna kill you, right? It'll kill this cell, right? Foreign cell. And then if you have a human cell that's infected, it's going to have that major histocompatibility complex type 1. But what happens is when it's infected, infected cells present far more activating ligand. So you get a whole bunch of these suicide by cop things. So there's a whole bunch of them. The natural killer says, okay, you're infected. We got to get rid of you, and it'll kill it. All right. So now another example of an innate immune system are macrophages, right? Remember, these are always on. These are always there. They're always working. These are always working, protecting you, right? And macrophages are like amoebas. I don't know if you remember from biology in like high school, but amoebas are single-celled organisms that kind of like flow around. They can change shape, right? They squirm around, and then they can eat things. Like this looks like a paramecium. It engulfs it. That's called phagocytosis, and it eats it, and then it digests it, so it kills it. Okay, so that's what macrophages do. But they do more than that. We'll talk about how they relate to the adaptive system in a second here. Okay, so the adaptive system examples. Remember, these must be turned on, all right? And it takes a while. It takes 4 days to respond, and that could be too late. But these are the big guns. These are like the heavy hitters, right? But they're not always working. All right, they only work when there's an infection. All right, you don't want to waste all the resources for these guys, right? Unless you need it. Okay, so some examples are cytotoxic T cells. You got these B cells, helper Ts, memory B cells, and memory T cells. So there's 5 different types. And I know you're going to get confused here. There's a lot of stuff here, but I'll go through it one at a time. So cytotoxic T cells are very similar to these natural killer cells. They're cops, right? So these are like, local city cops. These are like state police or something like that, right? They're cops also. So they go around and they check IDs, right? But instead of this combination of major histocompatibility complex type 1 and activating ligands, right, what they do is they look for major histocompatibility complex type 1s that are corrupted by a piece of the antigen. Remember, you have an infected cell. You take a piece of that pathogen and you place it on the major histocompatibility complex. So you place it on the ID, and when you place it on the ID, that's like a warning signal. So I messed up. I'm infected. All right. So let's see here. So when you place that virus antigen on the major histocompatibility type 1 complex, it makes it a phony ID or a corrupt ID. It says kill me. So then a cytotoxic T cell, which is like a natural killer cell, comes along, reads it, and says, okay, you're corrupted, right? And it sends these molecules called porphyrins, right? They make little pores, it like shoots little holes in it, and then the cell bursts and dies. It kills it. It's like a little machine gun. All right. Well, maybe not like a machine gun, but you get the idea. All right. So B cells make antibodies, and antibodies are proteins that bind to antigens that kill the pathogen. All right. So here's an antibody. It looks like a little Y. So you got a fork side and you got a tail side, right? The fork side is what binds to the antigen, right? And here you have an activated B cell. You got the antibodies on the surface. Now, some of these go to form memory B cells. We'll talk about them in a second. But some of these are modified to become plasma cells. Plasma cells are the ones that produce a lot of antibodies and release it. So now you got these free antibodies floating around. And they're like little missiles. They're like homing missiles, right? They go and bind to the antigen and they neutralize it or kill it. Okay? All right? So these are like little missiles. Think of them as like missiles. B cells, I remember antibodies. B cells make antibodies. Okay. Okay. So this is how antibodies work. All right. So here's a coronavirus. Okay. It's got the spike protein on there. Spike protein is what binds to the ACE2 receptor and folds it. Says, "Hey, I'm like one of those, angiotensin 2 molecules." And then the receptor says, "Okay, I'll let you in. I'll pull you in." So it pulls them inside. Right? So what the antibodies do, remember this, they were like little missiles, and this is the binding site, the Y side, the fork side it binds. It binds to the target here, which is the spike protein, and if it binds to it, well, it can't bind to the ACE2 receptor. Or if a bunch of these bind to the, around the virus, it can kill the virus. Right? And another thing, the third way is the tail side, the tail end of the antibody, right, is tasty for macrophages. So these guys here, these are the macrophages, right? They're all drawn a little differently, but they eat things. They're like amoebas, right? And if it's coated with these antibodies, then it's like icing on the cake, right? The tastiest part of a cake is icing, so it's like putting frosting on the cake, and then the macrophages come along and say, "Oh, yummy!" and it just chows down on these viruses, right? Eats them up. So let's review how antibodies work. Antibodies can bind to the COVID spike protein, preventing the virus from binding to the human cell, blocks it, right? Can't get inside, can't reproduce. Antibodies can bind to and kill the virus, okay? And antibodies coating a virus are like frosting on a cake, making the virus more tasty for the macrophages that then gobble them up. This process is called opsonization. You don't need to remember that. You're probably not going to remember that. That's a fancy term for this process. And, I don't know exactly how it works, but I'm willing to bet it has something like this. On these macrophages, they have receptors that bind to the tail end of these antibodies. So it's like, boom, they bind it, and they gobble them up. Okay. Kind of like this process here. All right. So that's M. And now another type of cell, so you have cytotoxic T cells, you have B cells, and then you got these helper Ts right here. So this is a macrophage, and they work together, right? So an innate immune cell works with an adaptive immune cell. And then macrophages eat things, so this is your pathogen. It breaks up the pieces here, right? Presents it on a major histocompatibility complex type 2. Remember, these are major histocompatibility complex type 1. Well, on macrophages, they're type 2, a little bit different, but they're really the same thing, right? They're an ID badge, right? And it presents the antigen on there, and when it presents an antigen, it's a corrupt ID. So that tells the helper T cell to get activated, right? Okay, there's a problem here, and what the helper T's do is they help the other cells and itself replicate or multiply, right? So this what they do is they release chemicals called cytokines. Remember the cytokine storm? And cytokines are chemicals that are signals that cause clonal proliferation. And when I say clonal, this makes clones, right? So you make a whole bunch of the exact same one here. Whole bunch of clones. So they help by saying, hey, you go make a whole bunch of, right? And then you make a whole ton of B cells. You make a whole ton of cytotoxic T cells. And then it activates itself. So you make a whole ton of helper T cells, right? So this ramps everyone up to make a big response, right? That's what helper T cells do. They help all these to reproduce. All right, so let's show what goes on here. Here's a little pathogen. It gets eaten, it gets digested, right? You take a major histocompatibility type 2, which is a self ID, right? And you combine it with that antigen, place it on the surface, and then a helper T cell comes along and says, "Oh, okay, we got a problem here, right? Right? There's a bag. So, hey, you guys, multiply." And then they make a ton of B cells, a ton of toxic T cells, and it activates itself. So you make a ton of helper T cells. All right. So then you get a big response. And then another thing is, like in this case here, so we said that activated B cells make these plasma cells, and these are the guys that pump out the antibodies, but they also make these helper B cells, and then also helper T cells are made too. So memory B cells. So there's memory B and there's memory T cells, right? Now, the adaptive system, they take a while to kick in. 4 days, they're the cavalry coming to the rescue. They're the big guns coming in, but they're slow. Take 4 days. And another problem is they don't live long. After the infection's gone, they say, "Okay, we're done," and they die. Right? No longer needed. Why waste the resources? Right? So they don't last long. You lose that protection. You get whacked again. It takes another 4 days. But what happens is these memory B and memory T cells live longer. Sometimes many years. Like when you get a vaccine for measles or something like that, right? You get it once in your life, and you're good for the rest of your life. They live forever until you die, right? And in some cases, they live only about a month. Like for the common cold or flu, they live only about a month. So you get this protection for about a month. They cause so what they do is they're all set. They're all ready to recognize the same pathogen again. They're like, okay, I remember you, right? You whacked this before, so and they'll recognize it right away, and they'll launch a rapid and immediate big adaptive response. All right. Right, so what do I say here? They cause a rapid and big response of B and T cells to a new infection. All right, so you're ready for another one. Okay, let's see here. So advantages of the innate system is that it's always on guard. It's always protecting. It's always turned on. Right? We talked about your skin, acid in your stomach, natural killer cells, and macrophages, and there's a bunch of others. All right, advantage of the adaptive system is that it's the big guns, it's the heavy hitter, it's the cavalry coming to the rescue. All right. The disadvantage of the adaptive system is that it takes a while to kick in, about 4 days to ramp up, which can be too long. By that time the infection may have gotten out of control to where a person is overcome and dies. Also, the adaptive system doesn't live long after infection is defeated. I mean, like, why bother wasting all this effort and resources making these, cells that you don't need? So there's no long-lasting protection from it. However, that's where the memory B and memory T cells come into play. These cells live long and remember the antigen from the prior infection. And when there is a repeat infection by the same pathogen, the memory cells cause a rapid and big adaptive response, right? That nips it in the bud, whacks the infection before it can take root and cause any problem, and you're barely going to even notice that you had anything, okay? So I want to give you an example of B cells, right? I'll explain B cells. Remember, B goes with antibody, so these are the guys that make antibodies, right? So I'll try to explain this whole thing with memory B cells. All right. So there is an antibody. Remember, it's a missile, right? The fork end is what binds to the antigen, right? And antibodies, what they are, they're protein molecules. They have binding sites on the fork side, right? Binding sites on this side, right? And the amino acids at the binding sites can be arranged in an infinite number of combinations so that any antigen can be bound. No matter what it is, there's going to be some antibody that can bind it, because you have an infinite possible number of B cells that each of them make an infinite different type of antibody with a little bit different arrangement of amino acids in the protein, and that changes the binding characteristics. So you can bind anything because you got infinite numbers of these, right? So it's kind of like a slot machine, or a random number generator. It just randomly arranges, in a different sequence, and then each one binds to different things. All right. Hopefully that makes sense. All right. Now, antibodies on B cells act like receptors. So these are antibodies on the B cells. They act like receptors. And if an antigen binds to an antibody, so here's the bad guy, right? And it happens, okay, so this B cell has antibodies that can bind it, right? So what that does is, as soon as that binds, it sends a signal, all right, to this B cell, say, hey, you're the one that works. It could be like 10 billion others don't work, but this one works. Hey, you're the one to reproduce. So that orders this one to undergo rapid clonal proliferation, and you make tons of these exact same copies to make the antibody that works and gets that antigen. All right. So the one that works, it replicates. It proliferates. It's called clonal proliferation. Produces a whole bunch of the exact same clones, right? All right, now, most of them form these plasma cells, and plasma cells are the ones that produce and pump out the antibodies, and these are like the missiles that go out and hunt the antigen, bind it, right? but some of them produce these memory B cells, right? So I say most activated B cells form the plasma cells that make and release antibodies, but plasma cells don't live long after infection is gone, right? I mean, once infection, why bother keeping these guys around? It's just a waste of your energy and resources for your body, right? Which is better spent on other things. So they shut these guys down, right? However, some activated B cells, right, form these memory B cells, and they stick around and live a long time. The activated B cells form memory B cells with antibodies on the surface to recognize a reinfection of that same pathogen. These cells live long and trigger a fast and big response, killing the pathogen before it can take root. So it bypasses that 4-day delay time. All right, you know, cavalry takes 4 days to ride the horses in. All right, well, you don't have to wait that 4 days, and you get a massive response. So this takes 4 days to happen. Well, this time it's right away, right? You know, here's the pathogen there. And, okay, all right, and the memory B cells kick in fast, all right, and they have a big response, much bigger. So each one is like, no, this is a big response, all right, and it's rapid. All right, so you can use this as a review. It's kind of like same thing I taught you, right? You can let's see here. I'll go through it slow. Yeah, if you want to review, you can examine this. You know, just pause. All right. Okay. Vaccines for viruses. Now, this is the meat of what I want to get to. I want to talk about the vaccines. So vaccines work by creating memory T and memory B cells that recognizes the virus's antigens before actual exposure to the virus. Right? Therefore, when there is a real exposure to the COVID virus, the memory cells are already prepared to make a rapid and big adaptive immune response, effectively defeating the infection before it has a chance to take root and develop. Right? So vaccines work by giving a prior exposure to the target antigen to be attacked without causing the disease. All right? Now, in the case of COVID-19 virus, the target antigen is a spike protein, which is both vital for virus production. Which is vital for virus production, right? Plus, it's vulnerable. Remember to be reproduced, viruses must bind to a human cell, be pulled inside, and then hijack the manufacturing machinery of your cell so that your cell makes more viruses. All right? All right? Now, if the spike protein isn't working, well, it can never attach to the cell, and if it can never attach to the cell, it can never get inside. Right? So that's why it's a good target. Now, by giving your body prior exposure to the spike protein, memory B and memory T cells will be all prepared for a real exposure of the COVID-19 virus and mount a big and rapid attack upon it, effectively destroying it before it can take root. So if you are exposed, well, you're all set, right? You know, you're going to get the jump on them, and you're going to whack them before they can the virus can do anything. So to give your immune system prior exposure to the spike protein, 4 types of vaccines are used. All right, there's actual virus, there's a viral vector, there's DNA, RNA only. All right, you don't have any other parts of the virus. These are viruses, right? You have target antigen only. All right. So we'll go through each of these. So actual virus. Now this is the traditional vaccine method that's been used since vaccines were invented. All right, and it uses a weakened or killed form of dangerous virus so that it produces the immune response, building memory, right, immunity without causing the disease because it's either weakened or it's killed. All right. So the bad virus, you get the real bad virus, but it's weakened or killed, so it can't cause the disease, at least in normal people. Right? We'll talk about there's a possibility where it can cause disease in immune-compromised people. All right, so viral vector, what you do is you use a totally different virus, a harmless virus. You modify it to carry the genetic code for the target antigen, in this case, the spike protein. For example, you splice the DNA for COVID spike protein into a harmless virus which can't hurt humans, which delivers the DNA so that the spike protein is made inside the human cells. Right? So here is a harmless virus, and here is a virus DNA, but instead you add this bit of DNA to produce the spike protein. Remember the spike protein? Spike protein's there. All right. So harmless virus that carries the genetic code for the target antigen. In the case of the COVID-19, it's the spike protein which binds to the human cell. All right. So DNA, RNA only. Use the mRNA for the spike protein, same like this, but you don't include all the rest of the DNA for the virus, right? You only include that little bit, all right? A little bit of DNA for the spike protein, all right? Encapsulate it in what's called a lipid nanoparticle. And what these are is a big fancy term here. Basically, it's a tiny bubble made of fat molecules, right? So lipid means fat, and so this is a bubble. It's spherical, not round. It's a whole, it's like a regular bubble, right? And these are fat molecules, right? And the mRNA is contained in there, and this is necessary because there's two reasons why this is necessary. One is, if it's mRNA and the COVID vaccines are mRNA, mRNA is much more fragile than DNA, right? More susceptible to breakdown, right? So this protects it, right? And also, this lipid nanoparticle is attracted to and fuses with the cell membrane, so it joins with the cell and then dumps this inside. I'll show you how that works down here in a second here. All right. So the genetic code for the target antigen is carried and protected by a fat bubble. All right. Okay. Then you make only this, the target antigen. Now the third option is, well, you make these outside in a laboratory, right? Usually they're made probably by recombinant DNA, which involves bacteria. All right, you splice in the gene into a bacteria, and the bacteria make these suckers, right? That's probably how they make it, right? The target antigen is a vulnerable but important part of the virus, right? Necessary for viral function, like the spike protein, right? So it's critical. This has to work, or else the virus can't reproduce. Or it can be a specific harmful toxic chemical produced by pathogen. So some pathogens produce specific toxic chemicals, right? And some of the vaccines for those, they target that toxic chemical. And if the antibodies bind to it, it basically deactivates that harmful chemical, so it can't hurt you. All right. So what I want to do is kind of show you what's going on for each of these. All right. So actual virus and viral vectors are similar because you're using a real virus. Right? The virus gets inside the human cell and it dumps out its genetic material, which is the whole thing for the virus, plus that little bit of DNA or RNA for the spike protein. All right. So because you get the whole package of genetic material, it goes through the ribosome, and you make all the parts for a virus. Let's say, here's a spike protein, here's a spike, and these are also parts. I just use a picture of geometric shapes to show like different parts. So you're making a bunch of this different stuff, right? A lot of this stuff is unnecessary. This is what you really want to get. So this is less specific. Now, since the whole DNA is inserted, a lot of virus parts other than the vulnerable spike protein will be made, and your body will mount an immune attack against all this stuff, which is kind of a waste. I mean, why bother with all this? I mean, this is what's really critical and vital here. All right. Now, with the DNA, RNA only, all right, happens is you have these little tiny lipid nanoparticles, which are little tiny bubbles made of fat molecules. All right. Now, the lipid nanoparticle is a tiny bubble made of fat molecules. The human cell membrane, so that, imagine this is a human cell, right? The outer membrane is made of what's called a phospholipid bilayer, all right? And so it's fat too, which is also fat molecules. Now, the fat attracts fat, and they can join together, right? So the lipid nanoparticle sticks to, because it's fat, sticks to the fat of the membrane of the human cell, and then they join, and then opens up, and it dumps that messenger RNA inside. And then when it's inside, it goes to the ribosome, and you make only the spike protein, only the target. So this is very specific, all right? And that's why these things work pretty well, I'm sure. That's probably one of the reasons why. I'm not sure all the other stuff going on. I'm not an expert on it, all right? But I'm willing to bet that's one of the reasons why. That's the main reason why. All right. Okay, the last one is you inject the target antigen itself. Right, so you just take, this is what you want to target. You inject this into the person. All right. Injected spike protein will activate the B cells and the helper Ts, but these have no way of getting inside the human cell, so the cytotoxic T cell response is never activated, and thus no T memory cells are formed. So you don't get the full protection from this method. You only get the memory B cells. All right, does that make sense? So this is what happens. Once a spike protein is inside, now in this case it never gets inside, right? So this part never happens right here, the cytotoxic T cells. Once you get that spike protein inside, and you got to inject the genetic material, right, in one of these ways here, all right, get it inside. All right. Then some of these are broken up, and then pieces are put on a major histocompatibility complex type 1, right? And this becomes a warning signal, flag upside down, warning, right? Warning, Will Robinson, right? And then that activates the cytotoxic T cells. This occurs only if these are inside. This is called the cell-mediated. It's got to be inside the cell, right? These are called humoral, which is outside the cell. It's like the fluid outside the cell, right? So some of these spike proteins get outside the cell, right? And then when they're outside, they're attacked by these B cells. And remember the B cell that recognizes it, right? It sends a signal, it attaches, you got an infinite number of these, but the one that binds it says, okay, you're the one who multiplies. And then you make, the exact same copies of that antibody which works. All right? Okay? So, and then another thing that can happen is the spike protein can be eaten by, especially when they're covered by antibodies. Remember, when they're covered by antibodies, it makes it tasty for macrophages, and they eat it, and then they digest it. They take pieces of it, present it on a major histocompatibility complex type 2, which sends a signal to these T helper cells. Now, the T helper cells release cytokines, which are chemical signals that tell all of these, tells cytotoxic T cells, it tells B cells, and it tells itself, more helper T cells, to rapidly multiply, right? So then you get a big burst, it's clonal proliferation. You get a whole bunch, and then you get a big response. So these guys play a big role because they help all of them multiply. All right. This is remember how antibodies work. The fork side is the binding side, right? And it binds to the spike protein, preventing it from attaching to the ACE2 receptor, right? So it cannot be pulled inside. And if a bunch of you can kill that virus, or the tail end is tasty, right? And it makes it tasty for macrophages. Here's a macrophage. It eats it. All right. right, so vaccine activation of memory cells. So vaccines give your immune system prior exposure to the target antigen, in this case the COVID spike protein, so that memory B and memory T cells will be prepared in advance to mount a rapid and big attack on COVID virus when exposed to the COVID virus, right? And these guys live a long time. Now, once a spike protein is made inside the human cell, parts of it are attached to the major histocompatibility type 1 surface surface proteins, which activate cytotoxic T cells that kill the infected cell, called the cell-mediated response. All right, I went through this already. Some of the spike proteins leave the cell where they are attacked by B cells. The B cells, which make an antibody that can bind to the spike protein, is rapidly reproduced in clonal proliferation. So you get a bunch of the proper B cell, then you get a bunch of the proper antibodies that work. When bound to antibodies, the spike protein is more tasty to macrophages, which gobble them up. The macrophages digest the spike proteins, breaking them down, but also presenting parts of the spike protein on a major histocompatibility complex type 2 surface proteins that present them to helper T cells. Helper T cells are then activated and secrete cytokines, which are chemical signal molecules, which tell B cells, cytotoxic T cells, and helper T cells to undergo big time clonal proliferation and rapidly reproduce. And that's how helper T cells help. Some of the T cells form memory T, and some of the B cells form memory B cells. Now, the B cell, cytotoxic T cells, and helper T cells of the adaptive immune system die off rapidly after infection is gone, because you no longer need them, and why waste the effort and resources and energy to make them, right? This is good because it saves resources, but it's bad because, the next time you get infected, it takes 4 days to activate this adaptive response again, right? And this is the heavy hitters. But the memory T and memory B cells live a long time, and they remember the COVID spike protein antigen and cause a rapid and big adaptive response that overwhelms the COVID-19 virus when exposed, right? So this is a preconditioning of the memory T and memory B cells that give immunity, right? So that's what vaccines do, right? You get these memory T and memory B cells all prepared, right? So when you see the real deal, it whacks it right away. So advantages and disadvantages of each type of vaccine. All right, you got the actual, you got viral vector, DNA, RNA, and you got the antigen. Right? So advantages, I'm not a vaccine or viral pathogen expert, but I don't know of any advantages of the actual virus over the new forms. These are the newer forms, right? Disadvantages: the immune response is not specific to the vulnerable target antigen. It attacks everything, right? Right, and so it can be like a waste of resources, right? And also, the weakened virus can still cause disease in immune-compromised patients. So if your immune system is whacked, or if you're on immune suppressants because you had an organ transplant or something like that, well, you got to be careful with a weakened virus. Now, the inactive virus is a killed virus. It is totally safe because it is killed, right? And it's not going to do it. But the problem with this is this often provides a weak immune response and may require several repeat shots, to get a decent immune response. All right, so viral vector and advantages: it's a weakened form of a harmless virus, so you get double safety. This virus is not going to infect humans and make them sick, right? Plus, it's a weakened form of the harmless virus. It's like double safe, right? And another advantage is it can use DNA, which is more stable than RNA. Remember, we said that RNA, in these lipid nanoparticles, they must be protected by lipid nanoparticles because naked RNA is very fragile. It can break down. If it breaks down, you're not going to make that spike protein inside the cell, all right? But DNA is much more sturdy. It's a double strand, double helix, um, and it's more stable, all right? Disadvantages of the viral vector. Well, you're not just making a target protein. You're making a whole bunch of other stuff, so you're wasting resources, right? And an example is the AstraZeneca COVID vaccine, which uses a chimpanzee. I believe it's an adenovirus, so it's like a flu virus for chimpanzees, which are harmless to humans. Humans cannot get this. Plus, it's a weakened form of that, so it's double safe. All right, it's a weakened form of a harmless virus. In this case, a chimpanzee flu virus that humans can't get. And what they do is they just splice in the DNA for the spike protein in there, right? And it's been shown to be between 70 to 90% effective, right? The 90% is a special regimen where you get a half dose and then a full dose. This is full dose, 70%, right? All right, and that's decent numbers for a typical vaccine. All right. So now the DNA RNA only, right? That's the one with the lipid nanoparticles. Advantages: has high target specificity. Only the spike protein is made, so only the spike protein is targeted, and it could be a possible reason for the high efficacy shown in drug trials. I mean, there's two like this. There's the Pfizer vaccine and Moderna vaccine, all right? And they've shown to have roughly around 95% efficacy, all right? And this could be a possible reason. Again, I'm not an expert on this, all right? There's probably all sorts of factors which I have no idea that are going on. I just know the basics here, and I think the basics are good enough for the average person, right? So I'm willing to bet that's part of it. It's high target specificity. You're only generating an immune response specifically for one thing only, the vulnerable necessary spike protein. So it's safe, right? Because you're not injecting any other DNA. There's no chance of getting a virus, right? I mean, all you're doing is all you're making is, the spike protein, right? No chance of contracting the virus because you can't make the virus, right, with only just a spike protein. You know what I'm saying? Right? Disadvantages. Well, you still get allergic reactions for some of the ingredients. I mean, you get allergic. The thing about this, allergic reactions apply to all these injections, possibly, right? Depending upon what type of ingredients are placed in there, right? So it's been shown that about 1 in 100,000 have got allergic reactions, right? So if you get allergic reactions to vaccines, and then have some caution here, right? Uses RNA. I'll put that number down. So let's see. 1 in 100,000. Uses RNA, another disadvantage, which is fragile and easily broken down, and it could be a possible reason for the cold storage. I mean, I don't know. It could be the lipid nanoparticles are fragile too. So I don't know, but I know that messenger RNA or any RNA, which is a single strand genetic material, is fragile and vulnerable, right? So examples are Pfizer, Moderna. They've been shown very high efficacy. This is like whopping high. Very effective, but they got to be, stored at cold temperatures. Pfizer is like super cold. Moderna, you can use a regular refrigerator, from what I understand, right? And then inject the target antigen. I don't know what advantages there are. I mean, maybe it'll work. I don't know. I don't know. But disadvantage is the protein never gets inside the human cell. So the cell-mediated response, right, is bypassed, and then the cytotoxic T cells are never activated. Therefore, the associated memory T cells are never activated. You only get a partial immunity established then. right? So this is less effective. And I give a caveat here, right? I'm not an expert. There are always a myriad of other factors that determine efficacy of each vaccine, right? I don't know these, right? I haven't studied. You know what I did? I used my background knowledge and I watched a couple of videos. That's it, right? To make this lesson. I spent like a couple hours, right? So I'm not an expert here. I don't know all these factors, and so you must rely upon the drug trial results. You got to go with what they say from their tests. All right, these are the numbers. All right, go with that. Right? I don't know the reasons why, all the detailed reasons for the efficacy. It could be they don't know themselves. I mean, there's another number of factors, and it's hard to figure out what's causing what. So here's a summary. The COVID virus primarily affects the respiratory system. The COVID virus obviously is pretty deadly in some people. In some people, they don't even know they have it, right? But it's a pretty deadly one, Its symptoms are similar to both cold and flu, right? The severity of symptoms can range from mild to severe. Severe symptoms appear to be due to an excessive immune response. Your immune system gets way too ramped up, and that causes damages to yourself, especially inflammation and death of alveoli, which are the tiny air sacs, millions of them make up your lungs. Right? But they're critical, right? Because that's where air is exchanged. That's where oxygen and CO2 are exchanged, right there. They're not working well, you're not breathing. You're gonna be, get asphyxiation and die. So viruses are not living things because they cannot reproduce for themselves. To reproduce, they must invade a human cell and commandeer its manufacturing system. And then the human cell makes more viruses. Okay? So viruses, they gotta attach to a cell, be pulled inside, and then, hijack the human cell's manufacturing system to make more viruses. A critical step in this reproduction is attachment to the human cell, and this is done by the virus's spike protein. The COVID spike protein binds to an ACE2 receptor, which is prevalent on lung cells. The spike protein is an ideal target for vaccines because it has a critical function and it is vulnerable to antibody attack. COVID vaccines work by giving your immune system a prior exposure to the spike protein so that memory B and memory T cells are all prepared to recognize the COVID virus right away when you're exposed. It's like, okay, I know who you are. I've seen you before, right? And then mount a rapid and big adaptive immune response against it, which otherwise would take about 4 days, which can be too late, right? So remember the adaptive system is your cavalry coming to the rescue. It's your heavy hitters, your big guns, right? But it takes a while, and that can be too late, right? In this way, the COVID virus is wiped out before it can take root because you're all set for it. You got to jump on them, right? You got to jump on that COVID virus because you know exactly what to expect and you're ready to jump all over them, right? That's how these vaccines work. They give a prior exposure, right? Get you all ready, right? To recognize it and mount the proper attack on it right away. Big, rapid attack. So these are the 4 types of vaccines for COVID-19. You have actual virus. I don't know if they're really working with actual virus and COVID-19, but I provide this because this is the classic means. You have viral vectors like the AstraZeneca. You have DNA, RNA only like the Pfizer, Moderna, and you have target antigen only. And I don't know who's doing that. So for more information, these please review the above here. All right, on vaccine types. Okay, that's it.